This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 154, recorded October 20th, 2011. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
How you doing, Alan?
Doing all right.
Nice day, isn't it?
Yeah, it cleared up nicely after buckets of rain, and things look good.
Yeah, we had a ton of rain this week.
Yeah.
As late as this morning, it was pouring. Beautiful. Beautiful now. Really nice. Also joining us from North Central Florida is Rich Condit.
Hi, fellas.
How you doing, Rich?
I'm doing well. This is good. I'm having a good time. Everything's good.
The weather's beautiful down there?
Yeah. There's a clear blue sky. It is 64 degrees.
Nice. Hey, it's warmer.
It was 47 this morning.
Wow.
Yeah. How about that? We could use some of your rain, though. I need some rain.
We have a lot of rain up here. Currently it's 61 here, which is, of course, Fahrenheit.
Yes.
Yeah,
But,
I don't know what any of that stuff is in Celsius.
I can tell you that it's 18 degrees Celsius, which I just read off my new iPhone 4S.
Ooh, you got an iPhone. Wow. I'm going to go out and get one this weekend.
First one.
I got to upgrade.
I've never had one, so I
You
figured
digging it?
this was a time. It's really cool.
Good.
I can do stuff
Glad you like
when
it.
when I'm at a faculty meeting and it's boring. I can do email.
Yeah. You need to download vaccines.
Vaccines?
Yeah, the app.
I got Health Map. That was cool.
There you go.
I should just go back through all the TWiV picks.
Exactly.
And get all these. You should get one, Alan. It's really cool.
Well, I have an iPod Touch.
Yes, that's cool too.
Yeah, so as long as I'm in range of Wi-Fi, I've got most of the iPhone features.
And it's getting so that you're almost always in range of Wi-Fi.
Yeah,
exactly.
Cool.
But I'm not paying the digital rates.
Well, so I know that the temperature at New York City today is 18 degrees. So there you go, all international listeners. Today we have two cool stories, and I want to begin with them right away because Rich has to leave in about 30 minutes.
Yeah, Rich's heart is nearly broken here. Actually, the meeting is right across the hall, so I got 40 minutes.
Great.
If you finish early, come back.
Okay, that's unlikely, but
yeah,
yes.
These student meetings go on. Before we start, a few weeks ago we said we're going to give away a free Drobo. Well, hardly anyone responded. Hey, guys and gals. Free Drobo. I'll make it simple. Go to bit.ly/DroboTWiV. Just fill out that form and you'll enter the contest. You don't have to do any tweets or anything like that, okay?
I probably can't enter, right?
I don't know. Just enter. Everybody enter. See, the thing is, if we get a lot of response, Drobo will go, oh, they have a good audience. We should advertise with them again.
Right, and you get a Drobo.
Yeah, and if you don't know what one of these things is, it is the backup solution. I've got one, and it's great.
How bad can it be? This is easy. There's like really 10,000 people out there. Do this, okay?
Yeah, there's a lot of you guys out there and gals.
Piece of cake.
It's good for us. We're not asking for any money, and maybe Drobo will give us some in the future so we can get better equipment and I can sound better.
Good for us, good for you. It's a win-win situation. Go for it.
Thank you. Okay, today we have two stories which just came out in Science, and they're both about how viral invaders get help from your gut bacteria. This is really cool. And
Thanks a lot, gut bacteria.
Oh, it is just amazing. The bacteria are letting us get infected with viruses. How about that?
I don't think they're doing it consciously. I think they're being duped.
I think they're not given any choice in the matter.
It's evolution. It just got selected this way, right?
I mean, the bacteria probably don't care. Unless we get killed.
Yeah.
Actually, if you wind up with gastroenteritis or something like that, that might even be good for the bacteria.
Or bad.
Bad.
You mean viral gastroenteritis?
Yeah.
It's a very interesting symbiosis. I don't know that the bacteria get anything from the virus, okay?
Well, that's what I was just wondering about.
But the viruses have clearly co-opted something out of the bacteria. But who knows? It remains to be seen.
Yeah, I suspect it would be bad for most of these. You get a viral gastroenteritis, it kind of cleans you out.
Yeah, but you get to spread.
Yeah, for most.
No, these are commensal bacteria. So
these are commensal bacteria. They don't have a problem with that.
But we're getting ahead of ourselves.
Yes, we're getting ahead of ourselves.
Yeah, well, you're not going to be here for the fun part, Rich, when we speculate. But
I'm already having fun. We're already speculating.
The first paper has to do with mouse mammary tumor virus. It's called Successful Transmission of a Retrovirus Depends on the Commensal Microbiota by Kane, Case, Kopaski, Kozlova, McDermott, Chervonsky, and Golovkina. And this is in Science. They're both in Science.
One of my immunology friends walked in on me yesterday while I was reading this paper, and I was telling him about it, and he said, that must be by Golovkina, or Golovkina, right?
I don't know
From
how to
Chicago. And he was really impressed that I was saying stuff like IL-6, IL-10.
Very good.
It was good.
Well, on Monday morning, Dixon comes in and he goes, "Oh, you have to look in the latest Science. There's a cool paper on microbiota." And I looked, and he was actually referring to a paper that came out a few weeks ago where they look at diet and enterotypes.
Oh, right.
So I picked that for TWiV this week, and then I saw these two papers, and I said, "This is TWiV," and that's how I found these.
Yeah, and actually, I found these independently, and I put a link in the Google Docs.
I saw that, yeah. And then,
Dixon would have liked this. It's got, germ-free and specific pathogen-free mice in it.
Yeah,
I don't know why he's not joining us. He's sitting outside, but we just did a TWiP. Maybe he's had enough talking. Not likely,
Not
right?
Not likely.
So I don't think we've ever talked about MMTV on this podcast, have we?
No, I don't think so.
Mouse mammary tumor virus. It's a virus of mice, as the name indicates. It causes mammary tumors, and it is often spread from mother to offspring via milk. And milk has virus in it.
And it's a retrovirus.
It's a retrovirus, right. So it integrates. And in fact, it's sometimes spread vertically by from mother to offspring in the genome, but also by spreading in the milk. So do we need to know anything else about this before we get into the paper? Is that enough?
I think that's probably enough.
Milk spread in the milk.
Basics.
Retrovirus. So basically these viruses establish chronic long-term infections. The mice are fine. They may get tumors from time to time, but these viruses exist in mice for long periods. And, of course, to do that, the virus has to somehow evade immune responses. And that's where this paper begins, because it's previously known that in order to persist in mice, this virus needs to produce a cytokine called IL-10. And, in fact, even more specific, it has to be dependent on the toll-like receptor 4. So it's called TL4-dependent production of IL-10. If you don't get that, the virus is cleared.
Right, and IL-10 is an immunosuppressive cytokine.
That's it, which makes sense, right?
Damping down the immune system, and the virus somehow trips this TLR4 to trigger the IL-10 and damp down the immune response so that the virus can persist.
That's right. And in fact, in their first figure, to illustrate this, they feed adult animals MMTV-laden milk.
So they infect them orally.
They infect them orally. They infect them when they're born, and then when they're adults, they still have the virus. But they don't have antibodies against the viral proteins because the virus has suppressed the production of antibodies by this TLR4-dependent IL-10 production.
Yeah, they actually take the adults and basically try and immunize them with MMT proteins, right, and see if they get an antibody response.
That's right.
And the mice don't respond.
Right, they appear to be tolerant to this antigen. They treat it almost as if it were a self, so they don't respond to it.
But they do respond to, say, ovalbumin, a different protein antigen.
Right.
Right. They're immunocompetent, but this particular antigen, they've got a blind spot.
Okay.
And as a control to this, that's interesting because it's specifically oral tolerance, because if they take mice at birth and inject them intraperitoneally, infect them that way instead of orally, and then come back later, those mice, I think, clear the infection, and they can mount an immune response to the antibodies. So it's specifically virus acquired through the oral route. And this, I'm naive to a lot of this stuff, okay? This oral tolerance is a big deal because we encounter a lot of antigens by eating stuff. And if we mount an immune response to all of those, we're going to be in big trouble all the time. So we have to have mechanisms to tolerate some of the stuff that goes through our GI system. So there's the immunity from the oral route. Oral tolerance is a special deal.
And
right.
Of course, we start when we're born, we're sterile, and immediately we start eating. Mm-hmm. And one of the first things we eat is mother's milk. Mm-hmm. So this virus has sort of taken advantage of that. Gets in there and makes it tolerant, makes the animal tolerant right away. It's very interesting. Now, this TLR4, toll-like receptor 4, it's one of the many toll-like receptors. There's also the Racaniello-like cytoplasmic receptors that all sense foreign proteins or RNAs. And TLR4 is known to sense bacterial lipopolysaccharide, LPS.
Right.
Right.
So that gave them some clues about what this virus might be doing, or at least what pathway to go down to figure it out. They say, well, it could trigger TL4 itself, the virus, or maybe it uses some part of the microbiota.
So for the otherwise uninitiated, LPS is a long polysaccharide attached to a lipid that exists on the outer membrane of gram-negative bacteria. So basically, a lot of bacteria out there are coated with this thing. So it's basically something that identifies bacteria.
Right.
So
And this comes up a lot in the innate immunity because the immune system obviously has been encountering bacteria for as long as there's been an immune system. And as a result, there's a lot of machinery in the immune system that's built around detecting and responding to lipopolysaccharides in various ways.
Yeah, responding or not.
Or not, yes,
right.
Depending on the context. In some contexts, it should not respond. For example, gut tolerance. In other contexts, if it's floating around in the bloodstream, that's probably a bad thing and it should respond.
Right.
So when LPS binds TLR4, this starts the innate immune responses. You have cytokines produced and cells are brought into the area where this interaction occurs. So it's a very important trigger for that. So they did an experiment where they basically treated mice with antibiotics to drastically reduce their gut flora and then see if they can respond to viral antigens. And what you find is that if you deplete the gut microbiota with antibiotics, the virus can the mice can respond to viral antigens.
Right.
Right.
So same experiment. You got the infected mice, but now they don't have any bacteria, and now they clear the virus rather than the virus persisting, and they can make antibody to it. I mean, so those two are obviously related.
Right. Treat them with antibiotics, reduce the flora. It doesn't eliminate it, but it reduces it substantially. And now the virus, the mice make antibodies against the virus. Okay, so that's one cool approach that suggests that the bacteria in your gut are important. The other one is to use germ-free mice and ask whether germ-free mice, which don't have bacteria in their gut, are able to produce antibodies, or even more than that, to be infected with the virus.
So do you know how you make germ-free mice?
Yeah, they say we produce them. I don't know how you produce them. I know you can buy them, and then you have to put them in your germ-free facility.
And you got to assay them all the time. So these have like no bacteria at all.
Right.
At least undetectable.
Right, as opposed to specific pathogen-free, which have some are missing. Some microbes are missing, but others are still present.
Right, and the germ-free mice, as I understand it, are difficult to maintain, not only to maintain germ-free, but also because they're not especially healthy in that condition.
I can imagine. That must be both expensive and difficult.
Yeah. Is that equivalent to Notobiotic?
I do not know. See, Vincent ought to be here.
Hey, Dixon.
He would know.
Vincent is here. Dixon, you mean?
Dixon.
Notobiotic.
I'm looking it up.
He said it's only certain strains of bacteria are present.
So that sounds more like
SPF.
SPF.
Yeah.
The germ-free are very difficult. I know Julie Pfeiffer, who's the PI on the next paper, collaborates with someone at her institute who has a germ-free colony. And I don't know how you would make them, because just treating with antibiotics isn't enough. Right?
Well, I read something about how birthing them by cesarean section to start with.
Yeah.
Okay, so they don't encounter any bugs from mom.
Just,
you do that in a sterile environment, and then obviously you have to rear them in a germ-free environment.
Yeah.
Anyway, so you make germ-free mice and you infect them, and what they find is that most of these germ-free mice don't pass the virus on to their offspring. They make an antibody response, and which clears it. All right, so no germs. Virus doesn't succeed at multiplying in these animals. It's not 100%. It's interesting. And they suggest that it might be LPS in the animal feed.
Right.
So I don't know what's in animal feed, mouse feed, but apparently there's some LPS which comes from bacteria, right?
Yes.
Because the mouse feed, even if the mouse feed has been sterilized, it probably was not produced exclusively from things that were bacterially sterile.
There you go. So the bacteria are broken, they're killed, but they still have LPS in them. Yeah.
Right.
So they in fact find some LPS in these germ-free females in the milk of some of the females, which presumably comes from the feed. So can you imagine doing this experiment and you go, what's going on here? It's kind of like one day it's this and one day it's that, and someone thinks of the feed. I don't know if I would have thought of that.
Oh.
That's why you have to talk to people. You go to someone and go, what's going on here?
Right.
I go across the hall with a cup of coffee and go, Condit, what's going on here?
And Condit would say, I don't know. Go away, don't bother me.
Yeah, that's right. He'd say, well, what about the feed?
Yeah.
Actually, that's the kind of thing that you probably need to talk to your veterinary staff about.
Yeah.
Because they'll often have a really good idea of, they obviously know what's going on with everybody's animals, and they're in touch with all these aspects of animal husbandry, and so the veterinarian who's in charge of the mouse house might be able to come up with something like that.
Right.
Well, you can imagine these guys would have been like totally focused on LPS. So if you've got something that doesn't fit your hypothesis, you would be thinking there must be some LPS in here somewhere.
Right.
Okay? And then you start wondering about the food and that kind of stuff.
All right, now
I spent forever on this table in 2C. For some reason, it this is one of these double and triple negative type experiments.
Oh, the mouse genetics things always take me an age.
I had a real problem with this, but it was fun because it was something that all of a sudden it clicked and everything made sense and it was okay. As a matter of fact, it wasn't until I got to the nice figure that they buried in the supplementary data that it all really started to make sense.
So the table Rich is talking about shows the different kinds of mice they tested and whether they're germ-free or SPF.
Right.
And
so
then,
now these mice that they're testing, they're now trying to figure out what is connecting LPS, since that's apparently involved, to viral pathogenesis, and they're looking at mice that are lacking different parts of this TLR4 IL-10 chain.
Right.
Right. So if you've got germ-free mice that don't have the TLR4 receptor, okay, then even if there's any LPS there, it's not going to be sensed.
That's right.
And all of those mice clear the virus.
Yes, eight of eight families, right. So that's compared to wild type 5 of 7. That's because of the LPS in the feed, right?
Right.
Right.
Okay, why don't you go on, Rich, because you got a handle on this.
Then they got this. Then they've got, I may fall flat on my face here, but then they've got mice that are missing MyD88, which is I would say, a ubiquitous intermediate in all of these signal transduction pathways. And
most of them, not all, but most, yeah.
A lot of them.
Yeah.
And in particular in the TLR4 pathway. And if you don't have MyD88, then you can't do any of this immune response, and the virus persists, even in germ-free mice.
Right.
And then they do specific pathogen-free mice, both wild type and MyD88, and none of those mice can clear the virus. So the specific pathogen-free mice are really no different than regular mice because they do have bugs.
Yeah.
So they've got LPS. They've just got a subset of the normal bugs. So there's a good correlation. In the end, there's a good correlation between bugs and an LPS receptor and whether or not you can clear the virus.
So MyD88 is an intermediate between TLR4 and what goes on in the nucleus in terms of transcription.
That ultimately generates all of these interleukins.
Right. And in fact, TLR4 can signal either through MyD88 or another protein called TWiV. Okay. The cool thing about this table is that in a MyD88 deletion, none of the families of mice are virus-free, even though they're germ-free.
Right.
Because apparently MyD88 is needed to clear the virus.
Right.
So even if you can't, if you don't have TLR4 stimulating LPS, the virus is not cleared because you need MyD88 to do that. And the other protein that can signal from TLR4, which is TWiV, they say that doesn't matter for clearance of virus. So this is kind of a side issue, but it threw me for a while.
Oh, yeah. Like I said, it wasn't clear to me until I got to this supplementary figure 7 that has a nice cartoon that shows all this. And what they show is that the antiviral immune response, they don't even talk about TLR7 in this, but the antiviral immune response, the thing that actually clears the virus infection normally, is done through another toll-like receptor, TLR7, that's also mediated through MyD88.
Yeah.
So if you don't have MyD88, you can't mount the antiviral immune response in the first place. So almost everything else is irrelevant.
Right.
Right.
Yeah, so there's two things going on here. One is that TLR4 interacting with LPS seems to be important for keeping the virus around, but clearing it depends on MyD88. And so MyD88 knockout, no virus-free families. Okay.
In the last few minutes, I have said TLR4, TLR7, MyD88, and IL6. It's incredible.
You're an immunologist, man.
No, not quite. I'm trying to learn the vocabulary.
Well, you could say it's a few other markers, and then you'd be.
Yeah, you think so?
Yeah. Okay, so now we see that LPS and TLR4 are important. So basically, they show next that LPS binds the virus. Okay, it binds the particle directly.
Right.
They can add it to virus, and even in a germ-free mouse, that virus can infect those mice just by the fact that LPS is present. So they do biochemical studies to show that LPS interacts with the virus particle, and that interaction allows the virus to infect mice, which is amazing.
Right. That is amazing.
And so I guess the idea is that you stimulate TLR4, which makes IL-10, and that allows the virus infection to proceed.
Right.
And only if LPS is present, which is a component of your microbiota, of course, can this happen. All right. So it's not that the virus can't infect, but it can't persist, right?
Right.
Without this immunosuppressive IL-10.
So what strikes me about this is that it's not just LPS. It seems to me that it's LPS, because there's LPS all over the place anyway. Mm-hmm. It's LPS in the context of the virus, it seems to me.
That's right. Yeah. It's the two of them together, yeah.
And so it's more complicated than just tweaking the TLR4 receptor. It's tweaking it with LPS that's in the context of the virus. I'm thinking there's probably components missing here, like co-receptors or or something like that triggers this tolerance.
Right, and because the virus does not induce a general immunosuppression, these mice still respond to other things, and in fact, it would be detrimental to the virus to do that because it's a good way to kill off your host too soon.
Likewise, LPS doesn't induce generalized immunosuppression.
But
it's LPS in the context of the virus.
Right, so LPS in the context of the virus is triggering this LPS tolerization pathway with TLR4 and IL-10, and something else, as you point out, is going on that is specifically tolerizing the host to probably viral antigens. Mm-hmm. So saying, hey, this is self, and then the host immune system assumes that for the rest of its life.
Tricky.
Rich, they do say that binding of LPS by the virus enhances its ability to induce IL-10 compared with LPS alone.
Right.
So yes, the virus is doing something, and there must be some other receptor or protein involved that remains.
I have
to
believe that these investigators must be looking for that.
Sure.
I would.
Yeah, it's fascinating.
Because that'd be fascinating.
Absolutely.
Yeah, and they make an interesting point too here, right at the end of the paper, that this isn't necessarily a mechanism that's going to be exclusive to the gut. So viruses that go through other mucosal surfaces, we have bacteria in our noses and in our throats and all other sorts of places. Something similar could be going on in those routes of transmission too.
Yep, absolutely. I'm sure people will be on it. I like this statement. They say LPS. So basically, they say LPS-induced signaling, of course, together with MMTV, drives a viral subversion pathway via IL-10 production that promotes viral transmission to successive generations. That summarizes it really well.
Yeah.
So I am really amazed or curious about how this evolved, right? I mean, who was there first, and how did it evolve this way? Could the virus infect but not persist, and then one day it picked up a little bit of LPS, and it just boggles the mind to try and go through the scenario.
Well, and this is not something that I would expect evolved with MMTV and mice.
No, this all has to do with MMTV taking advantage of the probably pre-existing oral tolerance. Oh, you're saying evolutionarily it goes back.
Evolutionarily, I think this probably goes way, way back. And when I first saw this paper, I thought I was just flipping through abstracts, and I looked at that. I said, wow, that's cool. I wonder how many other viruses do that. And of course, we're going to talk in a moment about exactly that question.
I think you're probably right. It goes way back. Yeah, it's an ancient thing because the viruses and the bacteria probably co-evolved, and
right, and the immune system.
And this probably happens in everything with a gut.
Right. So you get a gut, and you've got commensal microbes living in the gut from the earliest stages.
And so you have to have oral tolerance.
You have to have oral tolerance, and as soon as you have a security backdoor like that, something's going to take advantage of it, and here you go.
Mm-hmm. It's the safecracker thing again.
Yes.
So here we have an abetting role for a microbe in a viral infection, right? We did a paper on TWiV a couple of weeks ago where eliminating gut bacteria makes mice more susceptible to respiratory infection with flu. So there, the bacteria protect the mouse against viral infection, but at a different site.
Damned if you do, damned if you don't.
Can you imagine all the different scenarios? There's something with the skin. There's going to have to be something going on there with viruses that infect, maybe even mucosal surfaces like HPV.
Yeah.
It's just amazing. And just to think, it's a mouse virus that people are working on.
Yep.
Right.
That's not really translational research, is it?
No, that sounds pretty basic to me.
Sounds like curiosity.
Yeah.
Yes, that is curiosity-driven research.
And look what happens.
But it certainly has broad application.
Yeah, I think it's fabulous. It's a great story. I'd actually heard about this a couple of weeks ago. I was very happy when it came out. Anything else about this before we move on?
Well, I must say, when you first brought the paper to my attention, I was going, Vincent, this is immunology. All right? But I'm really glad you're making me do this because I'm learning a lot.
I hope we didn't confuse listeners with all the abbreviations. It can be daunting.
I think we did okay.
No,
we got to put this this cartoon from the supplementary figure ought to be maybe the image for the episode. The one on the left that shows the actual IL-10 thing summarizes it, because that really makes a lot of it clear, I think.
Will do. Yeah. You want to bow out now, Rich, before we go on?
No, I'm going to hang in there until the last minute.
All right.
Even if I have to bail out in the middle of this next story.
Got it. All right. So in the same issue, in fact, the following paper is a paper entitled Intestinal Microbiota Promote Enteric Virus Replication and Systemic Pathogenesis. And this is by Cuss, Best, Etheridge, Allen?
Preussers,
Frierson, Hooper, Dermody, and Pfeiffer. And I know there are some TWiV listeners in the Pfeiffer lab. So here we go. This has to do mainly with polio and also with reovirus. Poliovirus, which is, of course, the virus that Alan Dove did his thesis on.
Yes.
Remember?
He got a PhD for, right?
Yeah,
yes.
He did.
That's right.
He actually worked on poliovirus, as we have done in the lab.
I did.
For many years. And in this paper, they are using a mouse model for polio infection, a transgenic mouse model where you take the poliovirus receptor, identified in my lab by Kathy Mendelson, and you make a transgenic mouse, which was done by Rebao Ren in my lab, and those mice you can infect with polio. And so they begin by saying, when polio infects your gut, there's a lot of bacteria there. So what do they do? Do they interfere? Do they help? Do they hinder? So why don't we treat some mice with antibiotics and see what happens when you infect them with polio? So, and this is one of those experiments I wish I had done.
Yeah, it's a simple enough experiment.
Yeah.
Except you're trying to do plaque assays on feces. It strikes me that could be a problem.
There's got to be a joke there.
You got enough antibiotics in it. I guess maybe that's okay, but
yeah, you could filter it, right?
I guess.
A 0.2 micron filter ought to do it, because polio will go through.
You could flocculate it.
You could
yes, flocculate it. So they use these transgenic mice expressing the polio receptor. Now, when we made these mice, we used to feed them polio, and nothing ever happened. You could inject the virus in their brain or IP or intravenously or in the muscle, they would get paralyzed. But if you fed them, nothing ever happened. And many years later, Satoshi Koike in Japan found that if you don't take away the interferon system, nothing happens. But when you take away receptors for interferon genetically, then the mice become orally susceptible. So that's what they use in this paper.
So these are mice that are transgenic for the poliovirus receptor gene, the human poliovirus receptor gene, and they have had their interferon receptor gene, one of their interferon receptor genes, knocked out genetically.
That's right. So they don't respond to interferon, and apparently that's important in the mouse gut. Now, all of us, of course, have interferon receptors, as far as we know, and we can get polio. So our guts are clearly different from the mouse gut. And why that is, we don't know, but that's another story. Right. So basically they find if you treat mice with antibiotics, very similar to what was done in the previous paper, untreated mice succumb more often than mice treated with antibiotics.
Doubles the mortality.
Doubles it. So you get rid of the bacteria, and polio doesn't kill them as well, and it also doesn't replicate as well in their gut. And this is only by feeding the mice.
Right.
If you inject it, say IP, no difference, antibiotic treated or not.
And they have a cool control where they take the antibiotic-treated mice and recolonize their guts with what? Antibiotic-resistant bacteria,
yes.
Right?
Yes.
Yeah.
And when you do that, you're back to the situation that you would have. It's like the untreated mice.
Right. Now, just measuring virus in feces, they found wasn't very informative. So they actually do a very cool experiment, which happens to be ancient. Well, maybe not ancient, but many years old. And that is you can grow poliovirus in cells in the presence of a compound called neutral red. And the neutral red gets packaged with the RNA genome in the virus. Okay, so if you take neutral red virus and you shine light on it, it knocks out the infectivity.
Yep.
So that's how you can tell that what comes out of a mouse is what you put in or if it's replicated.
Right.
Because really, when you think about it, you feed mice polio and then you take the feces and do a plaque assay. You don't know if that's the inoculum or if it's replicated.
Right. So you might just have a high background of unreplicated virus that just passed through the gut.
Right. And they had some issues, and they used the neutral red virus to show that, in fact, if you deplete the microbiota, the virus doesn't replicate as efficiently as in wild-type mice. So that we don't have to actually talk about that, but I thought it would be an interesting aside for our listeners because it's a really clever trick that you can find in the literature 40, 50 years ago.
Yep.
And some old techniques are sometimes still useful.
I remember people using that in the lab when I was in your lab.
Yeah. I think it was Kathy Mendelson around.
Kathy was not there. Who was doing... I think Sa might have been doing something. Sa Liao was doing something where she needed to distinguish inoculum from replicating virus and did it that way. And she would have her plates covered with tinfoil and everything in the incubator.
Yep.
So it could grow without light.
I remember when Kathy used to make her neutral red virus, she would turn the lights off to check the cells, see if they were infected. If someone happened to come in and turn the lights on, she would scream at them. Turn the light off, you're going to kill my virus. Anyway, so getting rid of the microbiota for polio as well seems to harm its ability to infect. Okay.
Right. So the untreated mice or the recolonized mice die. The antibiotic-treated mice survive.
Right.
And it's and the virus that you're getting out of the antibiotic-treated mice is mostly what you put in.
Right. Okay, so then they get the idea that some component of the bacteria might be involved. So they have an assay where they simply incubate poliovirus in vitro in the absence of cells at various temperatures, and they add stuff to it, and then they quantify the infectivity. And they show basically that virus, if you treat virus with just a buffer, you lose infectivity. But if you incubate them with feces of mice or germ-free feces or components of bacteria like LPS, you can restore or enhance the infectivity. So LPS is, in fact, one of the most potent enhancers.
I can think of times when I've been so frustrated I wanted to defecate in one of my experiments.
Hey, it would have helped.
Yeah.
God.
So let's see.
That's pretty amazing. You can actually mix the virus with poo.
And it makes it more infectious.
And it makes it better.
Yeah. So here, polio incubated with PBS, feces from antibiotic-treated mice.
Are not improved by that, but yeah.
So if you incubate the virus with PBS, feces from antibiotic-treated mice, and germ-free feces, you lose viability. But if you incubate an untreated or germ-free feces supplemented with bacteria, you improve the viability. Bacteria don't have to be living, and LPS turns out to be one of the best stabilizers, if you will.
Right.
They show the virus actually physically binds LPS, and that's about it. They don't go beyond that. We don't know if it's binding, a particular receptor in the gut or what. So that remains to be seen. What LPS binding is doing. Maybe it simply stabilizes it, right?
But it seems to improve the virus's ability to get into cells. And an important distinction here: we're just looking at virus getting into cells. It's not to do with the immune system.
Right.
So whereas MMTV was using the LPS as a way to bypass the immunity, in this case, poliovirus appears to be using it as just an entry assistant.
Or a stabilizer, maybe.
Or a stabilizer.
Yeah. So maybe you get more virus infectivity and therefore a better infection, and that's all it is. It could be as simple as that. Right. So I like their conclusion. Our work implies that antibiotic-mediated microbiota depletion can have antiviral effects, although we do not advocate the use of antibiotics to prevent viral disease. I'm glad they added that.
Mm-hmm.
Yes.
Otherwise,
and they also did this with another virus too.
Yes.
So after finding this with poliovirus, then they looked at reovirus. So they're another enteric virus, and they did a similar set of experiments, antibiotic treated versus untreated mice, and looked at pathogenesis in these two, and you find once again, and did the follow-up type assays. And once again, the virus appears to use LPS as some kind of stabilizer or entry type assistant.
Here, of course, Rio is a natural virus of mice.
Right.
So one could argue that it's kind of is an important experiment to include to show that it's not a specific phenomena to polio.
Right.
I like the phenotype of Rio virus
Yeah,
infection.
you don't often see pictures of poop in scientific papers.
Right.
In the description.
Yes.
Feces from untreated mice were yellow, oily, and hardened, typical of biliary obstruction from viral replication and damage. Excellent.
And with that, fellas, I'm out of here.
Hey, we did the two. Very good.
We did the two. Not bad.
Thank you,
Sorry
Rich.
to miss the rest.
Thank you very much.
Have a wonderful time.
Right.
I will listen to you on TWiV.
Have a good meeting.
Yep. See you.
Bye.
Take care.
Okay. So, two papers, two different enteric viruses, or three. Their infection is facilitated by enteric microbes.
Yes, and by different mechanisms.
One involves immunosuppression, right, to allow persistence, and the other may be just enhancement of infectivity.
Right.
So I think a lot of people are now going to be looking at this, right?
Yeah, I think this is going to turn out to be a general story that viruses have, particularly enteric viruses, they're in this milieu where they have all these bacteria around them, and they're adapting in different ways. And polio and Rio are pretty much guaranteed to end up in this environment with this LPS floating around. They might as well use it for something.
Yeah.
And they may get some kind of advantage, maybe if they had to stabilize themselves rather than relying on LPS, it would require more energy, or they'd have to have a different shaped capsid, whereas LPS provides this. Or it could even be that this helps to concentrate the virus somehow in the areas where it's most likely to be able to infect.
Do you think that the stabilization by LPS years ago allowed this to be a site of infection?
Maybe.
Or did it happen later?
Or it may have been the other way around, that this was the site of infection, and it's an advantage that LPS is kind of an indicator, hey, you're in the intestine. It's time to infect a cell, rather than trying to infect cells in the esophagus, which wouldn't be productive.
So we have a gut microbiome which can influence virus infection. We know there's a skin microbiome, right? Where else are microbiomes in and on us that could participate in virus infection?
Any surface, certainly any mucosal surface. Your skin, of course, is contiguous with your mouth and your gut, so you follow all that through.
How about the lung? Is there a?
I would assume there is. I don't think there's been a lot of work on it.
I see there are calls for proposals from the NIH to study the lung microbiome. Yeah, because it's not sterile, right? Because you're breathing in.
I can't see how it would be.
Yeah, so that could influence infection. I guess there's an eye microbiome.
Yeah. Yeah, there are penile and vaginal microbiomes.
It's amazing. It's very cool.
Just any surface on us that's in contact with the world.
A mouth microbiome, yeah.
Absolutely, that's huge.
And a number of viruses infect there.
Yeah.
Oh, wow, this is going to be fun. And look at this. If we had eradicated polio 10 years ago, this work would never have been done.
You'd have to use some other enterovirus.
I guess that the field would have turned to another entero, and there are plenty of those that do replicate in mice. But the polio system is so compelling because you have so much information and so many reagents. You have this mouse, you have the genome cloned and sequenced.
Yeah, and you have a half century of classical virology that's been done on it. It's very hard to take an established system like that. It would be like if all of a sudden we decided, I don't know, we're going to eliminate mice. And now all work that's been done on mice, we just have to stop. You have to get rid of all of them. You have to switch to rats or pigs or whatever else, and you wouldn't be able to refer to this vast resource of the model that you'd built.
Yeah. So kudos to the Pfeiffer lab for continuing to work on polio.
Yeah.
In the face of difficult times, it's not easy to get funded on polio.
No, I don't imagine so.
There is some sentiment on study sections that, it's going to be eradicated soon.
One of these days.
Yes, as you know.
Yeah.
Okay, nice papers.
Yeah.
Let's do a few emails.
Sure.
First one is from Gabriel, who writes, I just finished listening to TWiV 152, in which you spent quite some time discussing the death of Steve Jobs the past week. Though this mention is certainly well-deserved, I thought it was an oversight not to make any mention of the outstanding immunologist and fellow New Yorker Ralph Steinman, who also passed away due to the same disease that same week, only days before being awarded the Nobel Prize for the discovery of dendritic cells. I know you guys are not the greatest fans of immunology, but I think that it deserved at least a mention, especially given the huge importance of DCs to the way our immune system senses and responds to viral pathogens. I don't
Absolutely correct.
I agree. We should have mentioned it, but we are fans of immunology, don't you think?
Yeah, absolutely. We, I have the greatest respect for people who work on immunology, and it's just they've discovered so much that it's often hard to get my head around all this stuff.
Well, we too, from time to time, cover immunology, and it would be good to have someone here from time to time who knew more, and maybe someone will show up. Yeah, we should have mentioned Steinman and Bruce Butler, who also co-won the prize. But in fact, we are going to do a show on Ralph Steinman in a couple of weeks. Jeremy Luban worked in his lab.
Right.
He's going to be in New York in a few weeks, and he said, let me come by and I'll reminisce about Ralph. I'll tell you what it was like to work in his lab and what he was like.
Outstanding.
And what he did. So Jeremy understands all that. So we'll do it then. But that was quite a story that he was awarded the Nobel Prize and had died a few days before, right?
Yeah, they don't award them posthumously, but they had already made the decision. They just hadn't announced it. And then they announced it, and then they found out that he'd died a few days before. And I gather the Nobel Committee said, well, we would look like total jerks if we rescinded it now, which they would have. Yeah. Not that stopped them from some of their earlier decisions, but in this case, I think they made the right call.
Yeah. So, yes, you're absolutely right, Gabriel. We should have mentioned it.
Yes.
And we will make up for that in a couple of weeks.
Yep.
All right. Alan, why don't you take the next one?
Falaye or Falai. Sorry, I've butchered your name. I've probably just mispronounced it 3 times, so I'll stop now. Anyway, he writes, Hi, TWiV crew. I am Falaye Temetope from Nigeria. I've finished a master's degree in virology in Nigeria and will be convocating in November 2011, which I guess means graduating.
Sounds like it.
Though I've been offered a PhD position in Nigeria, I'd love to experience education in another part of the world. I've been searching for what to do for a PhD, and in this light, I went to this meeting in Sapporo, Japan, IUMS. It was mind-blowing. It was my first time at an international meeting and a virology meeting at that. I learned a lot and just assumed the TWiV crew would be there. I was, however, not very happy to not see you guys there. Well, they didn't offer us a ticket. We absolutely would have gone. At the meeting, I learned about Saffold virus for the first time. Saffold virus is a human cardiovirus, first identified in 2007 by Morris Jones and his group in California. The virus was isolated in 1981 from an 8-month-old female child with fever of unknown origin. On returning to Nigeria, I downloaded everything on Saffold virus listed in PubMed and am churning through it all. I'm surprised to see the likes of Nick Knowles, Eric Delwart, Howard Lipton, Don Ganem, Joseph DeRisi, and Nathan Wolfe already neck-deep in the field. What was more surprising was the fact that I have no memory of Saffold virus being discussed on TWiV. The way you guys covered XMRV has built in me a level of trust in your judgment as a team, and I will so appreciate you guys discussing Saffold virus on TWiV. Should I venture out to work on for a PhD Saffold virus in Nigeria? Please, I'd appreciate it if you could, in addition to your opinions, add those of David Baltimore, Carla Kierkegaard, Ian Lipkin, and any other individual you feel is well positioned to inform my choice. You guys have constantly been a source of strength and support to me. I'm glad to inform you that I finished top of my MSc class. Congratulations. And I have to confess that TWiV and Professor Racaniello's virology lectures played a big role in my education. You guys taught me a sizable chunk of all I know today about virology. Thanks, guys, for all the effort you put into educating me and everyone out there who in one way or the other has benefited from TWiV. Look forward to hearing from you. And I gather he sent some pictures.
He sent some pictures from this meeting, and he has one with David Baltimore. And others with other virologists. So he said, feel free to post, so we'll post them.
Yeah.
And Saffold is an interesting virus.
Aren't they all?
You know, you're right. Every one of the millions and millions of different viruses out there. And we should do a show on it, so we'll put that on our list.
Yeah.
There is a review about to come out in Journal of Virology on Saffold, actually.
Oh, great.
Fallet, and you might want to look out for that. So thanks for that. The next one is from Dan. Hello, I'm a second-year medical student and was turned on to your podcast by my medical microbiology professor. All right. I love listening to your shows because they integrate the often dry, tedious concepts and bugs that I'm learning about into relevant, amusing, and easy-to-follow stories. You make learning entertainment. What did someone call it once? Edutainment.
Edutainment, yes.
A few episodes back, you mentioned that you were looking for a Virology for Dummies style textbook for amateur enthusiasts. While not specifically virology-oriented, I found the book Clinical Microbiology Made Ridiculously Simple by Mark Gladwin and William Traddle to be a great resource. It takes a humorous and lighthearted approach to a fairly complicated and, as I've said before, often tedious subject. Through humor, cartoons, mnemonics, and other imaginative study tricks, it's a great way to understand the basic concepts of medical microbiology. Since I've been completely immersed in all things medicine science for the past few years, it's hard to judge just how ridiculously simple the book may appear to a complete layperson. However, for someone with basic science knowledge, e.g. knowing the difference between RNA and DNA, and trying to get more insight into the world of microbiology, I highly recommend it. The best part about this book is its knack for juggling basic concepts with just the right amount of detail without overwhelming the reader with technical jargon or complicated, seemingly random facts. It is able to integrate and link concepts without coming off as a bug parade, as most micro courses books often do. Besides a few chapters on viruses, classification, life cycles, structures, specific viruses, etc., it spans concepts of microbiology from differentiation of gram-stained organisms to specific organisms to parasites, fungi, prions, and even pharmacology. Definitely worth $25. Please keep up the good work.
Cool.
Okay, thank you, Dan. We'll post it, and people can check it out. Alan.
Ricardo writes, hello, Vincent and the rest of the TWiV gang. Congratulations for the 3 years. I haven't written much, but you can be sure I'm there listening every Monday on my driving to the university, talking back to you as if I was there. One of your listeners said it seems like you're our friends. Well, it sure feels like it. I can see a tertulia about virus with all of you sitting at a cafe table. A tertulia?
I don't know what that is.
Tertulia.
Do you know? You don't know? Let's Google it.
It's some kind of event, I think.
It's a it's a cafe, bar, restaurant, northern Spain.
A tertulia. Oh, okay, a social gathering.
Okay, yeah, got it. And they named
in Spain or Latin America.
Nice.
With an academic overtone. Okay.
I've learned something there. Huh?
Yes, tertulia. I'm giving a little bit back with this video once again. You might use it on a public TWiV event while you wait to start the show so people can see a little bit of the TWiV history. Best regards, Ricardo, who is at the Fernando Pessoa University in Portugal. And he sent a video. It's a computer-generated video of a timeline of TWiV in the episodes starting from number 1.
All 150.
Yes.
Thank you, Ricardo. All right, the next one is from Jeffrey, who writes, Doctors and Alan, I don't remember mention of a PhD for you. Well, we did today.
I do have one.
Yes, and he got it in my lab.
That's right.
So, yes, Alan should be included there. PhD, in fact, working on poliovirus.
That's right.
I don't follow these matters closely, but I have a point that I feel probably confuses a lot of people and probably contributes heavily to rumors of continuing U.S. offensive biological weapons programs. As pointed out in episode 151, there are now quite a few defensive programs in place in the U.S. As part of their research, some of these programs actually do weaponize biological agents. This is not because they are necessarily looking for the next best weapon, nor because they think defenses against these will be of direct use in the future. It is rather because weaponizing, indeed any genetic manipulation, has unpredictable, at our level of technology, system-wide effects, and they are creating these agents to gain insights into how weaponizing might make biological agents more or less susceptible to established defenses. Sure, they keep these creations around for future study, and that is, I believe, what causes many rumors of weapons development. However, the difference between defensive and offensive development is mainly in quantity. A few vials of a weaponized strain, while dangerous, is not a weapon. A few tons of micronized smallpox or anthrax ready to be distributed to warheads is a weapon, as was the case before the disbanding of the Soviet Union. To the best of the public's knowledge, the U.S. is not stockpiling biological weapons and is not therefore engaged in any offensive programs. This does not mean the U.S. does not have some gems in a freezer somewhere that they are prepared to produce in quantity should the need arise, but that is not an offensive program.
Good point.
All right, thanks for clarifying that.
Yep.
And the last one.
Okay, Timothy writes, Dear TWiVers, I'm responding to your discussion about MD PhD training in your email-only episode number 151. You got it mostly right, but there are some nuances that were missed. While MDs can do basic research and PhDs can partner with MDs to bring clinical translation to their work, we clearly need people at the interface who do both for a more seamless transition. The two pathways for a person to reach the point where they can take care of patients and conduct bench science are, as you said, 1, the combined program, or 2, an MD who trains extensively in the laboratory. Many of my colleagues that have taken the latter route received their laboratory training during a subspecialty fellowship years and stayed under their mentor's wing until they were able to procure their own K and then R-level grants. Those are different designations of NIH grants. They learned to do science by apprenticeship, however, without the rigorous oversight of a thesis committee or completing the academic requirements of a PhD training program. I believe that the PhD training equips the individual with a more robust tool set for conducting research, and ultimately they're better poised for success. There's a downside, of course, in that the training is long during years of being a student and going further into debt. I myself essentially was in school for 17 years after high school before my first real job. Of course, talented individuals will be successful no matter which route they take. I do want to emphasize how important it is for there to be people that do both, because they can get things done or see insights that two people from different spectrums can't appreciate. And Tim is, as you probably figured out, an MD PhD, and he is at University of Cincinnati.
Yeah, it's good.
That's a good point.
We didn't get that advantage as we talked about it previously, so thanks for that. Tim is also the author of This Week in Pediatric Oncology. TWiPO.
Great.
So check that out if you want to learn about cancers of kids.
Yeah, and I think for the MD PhD discussion, we're going to bring somebody on who is an MD PhD and can talk about specifically their trajectory, right?
Yeah. We have one coming up in a few weeks, and everyone has a slightly different view, but we're getting a bunch of different ones, so it's good. If you are out there and you have an MD PhD, add to the conversation for sure.
Yeah.
And let me just do the last one, which is short.
Yeah.
From Georgianne. Has Ela Singh made a statement about whether XMRV is still a player in prostate cancer? I'm surprised I haven't seen a statement from her regarding this issue. Have I missed it? I would think she would go with the latest studies that show XMRV is a contaminant in prostate cancer. Thanks. Well, I was at a meeting in Chicago called ICAC, and I heard Ela Singh gave a talk, and she is still detecting some antigens in prostate tissues using antisera that she prepared against XMRV. Now, she says she realizes that XMRV itself is a lab contaminant, but she's leaving open the possibility that some related viruses might be involved in prostate cancer.
Or some other antigen.
Some related antigen, yeah.
Because it's not necessarily
sure.
an infectious virus antigen that she's detecting.
Yes, unfortunately, all she does is she takes tissues and sections them and probes them with antibodies.
Right.
So, yes, it needn't be a virus antigen that's there cross-reacting with the antibody. And she admits that her antibodies are a bit dirty. They do detect cellular proteins. So she said she's making monoclonals at the moment to XMRV, and we'll be using those to look at her tissue sections. So I don't know. I'm very skeptical that a retrovirus is involved in prostate cancer. As I've said before, to think that a contaminant would point you in the direction of a bona fide retrovirus infecting humans seems unlikely.
Yeah, it would be completely random.
Yeah.
That you'd get a useful lead out of that.
Exactly. It just seems weird. But so she is still pursuing that. She's doing a lot of work. And in fact, now she has moved to New York City. She's here at Mount Sinai.
Oh.
And we'll be hearing more from her. So she still thinks there is some connection with some infectious agent. So stay tuned. She hasn't published any of that yet. So I don't want to talk about it, but I'm sure we'll see it eventually.
Great.
All right. Let's do a few picks of the week.
Okay.
Alan.
Well, my pick is I actually had something else, and then this discussion got more interesting. So my pick of the week is this post that Seth Mnookin put up on his blog. He's calling it SciWrite Labs, SCIWRITE Labs, and it's an experiment in communication between scientists and writers. And Vincent and I are participating in this. Vince, you just had a post up that was quoting you, and we're now talking with Seth back and forth over email, and this all grew out of a discussion about fact checking for scientific articles, and it's an interesting debate / discussion about when and under what circumstances should science journalists run their articles or pieces of articles past sources or independent scientists to check the facts. And this is a discussion that actually grew out of TWiV from your episode in Chicago, because it was something that Trina Sideros said that really brought this about.
Right. And so I've picked a blog called Take as Directed, which is written by David Kroll, who did the original post about Trina's statements on TWiV in Chicago. He did a very nice blog post a few weeks ago, and there were lots of writers commenting there. So Seth took off on that, and he's continuing it. But Take as Directed is by David Kroll, who is a professor of pharmacology in the U.S., and he's very interested in communication, hence his participation in this. So it's a very good blog, covers a wide variety of topics, not only pharmacology, but professional development of scientists and so forth. So I thought I would add that in because it's relevant to what Seth is doing there.
Yep.
What do you think about Seth's SciWrite Labs?
I think it's a great idea. I think this was a discussion that obviously the time was right to have it, and Trina kind of touched it off. And now there's a lot of discussion back and forth between scientists and writers, which is always good. Talking about, obviously there's this tension between journalistic independence and making sure that the facts are carefully checked by somebody who knows what's what. And it varies from publication to publication, and depending on your on the topics that you're covering. Obviously, there are some topics where it's going to be much more technical and much more difficult for a general journalist to get everything right. And so I think it's a really good conversation about where do we find that balance. Because I'm sitting right on the fence, really. I see as a scientist, I see that it's crucially important to get the correct information out there to the public. As a journalist, I see that it's crucially important for the press to be independent from from influence by the people that they're covering. And that's scientists or politicians or business people or whoever they're covering. So you can't just send your articles to the researchers you're writing about and have them vet them and rewrite what you wrote. I don't think that would be appropriate. I think that would compromise the independence of the journalistic coverage.
Yeah.
But at the same time, journalists just writing it up and not bothering to check any facts is not really the way to go either, because obviously a lot of wrong information gets out that way.
Yeah, I think Trina's point was, as you know, to get someone else to check it, not the person you're writing about, right?
Ideally, yes.
Which is you have said in this discussion is not always possible.
Yeah.
You got to find a Racaniello somewhere.
Exactly. It is you made the assumption that all researchers behave as you do and take requests quickly and reasonably, and that's just, unfortunately, it's just not the case. If I send something to a researcher and say, "Hey, I'm writing this story about somebody else's work. Can you check it over?" I'll never hear back.
Yeah, I did make that assumption, and it was an error. I didn't realize that. But, it's worth trying at least, because you might be able to identify a number of people in different fields who are cooperative, right?
Yeah.
They're out there. So this is an interesting conversation, and check out both of these blogs for more on this. You'll see it's very testy at times because people are sensitive about what they're doing for reasons that Alan has just said.
Yeah, there's a lot at stake on both sides.
Yeah,
but I think overall it's a case study in how to have a discussion on the internet.
Yeah, no. So
I mean, there's been a little bit of this and that and some silly things said, but the overwhelming majority of it is a bunch of people who are genuinely interested in doing the right thing and figuring out what the right thing is to do, and they're just having this honest intellectual discussion.
Now, there are a few people who say, "No, I don't check," and that's the end of the story. And that's not really useful, but there are many. And those people kind of tainted my view of the whole conversation. Those are the ones that stand out in my mind when I think back on it, and
right.
not the ones who are reasonable, unfortunately. But the whole issue is very interesting. The whole issue of science writing is very interesting because it's difficult, because you have often people who are not scientists writing, right?
Yes.
So you are unusual because you are one, but not
right.
everyone is one, and not every science writer needs to have a PhD, right?
No, and in fact, for general publications, most of what they're going to be covering is going to be stuff that an ordinary person ought to be able to understand from the outset. And ought to be able to get the straight story direct from the researchers and not have to do a whole lot of back and forth. But there does need to be a lot of care about getting the story straight, because when you screw it up, it can have real implications.
So your view is that a good journalist should be able to talk, say, to a string physicist and inquire enough so that they can write a reasonably informed study, which is correct, right? They don't have to know the field in order to be able to do that.
Yes. Now, they're not going to write a description of string theory that a physicist would read and say, "Oh my gosh, I'd never looked at it that way."
Right.
They're going to write a summary that a physicist would look at and say, "Well, yeah, doesn't everybody know this?"
Right.
But the average person, the average biologist like me, would pick it up and say, "Wow, I never understood this string theory thing, and now I know something about it."
Mm-hmm.
So a good journalist, a good general journalist, science journalist, should be able to do that with any topic, but it's a question of talking to the people involved and really just kind of going around and around with them about, well, does this mean this or that, and really just nailing it down in your own mind before you write.
So Seth teaches a graduate program in science writing at MIT. So
right.
Are those some of the skills that you would be taught in a course like that?
Yes, and in fact, I taught in the graduate program of the journalism school at NYU for a number of years before I moved out of the area and the commute got too extreme.
I remember I came and...
That's right. I had you come and talk to them at one point, and they came to your lab and interviewed the folks in the lab too.
Right.
So yeah, there are a lot of exercises like the one that we did, having scientists talk and having people interview scientists as a practice, and also lots of discussion about how do you figure these things out, and how do you set up an interview, and how do you really kind of delve into a topic like this?
I'm sure it's a skill because, you may not understand something. Someone's talking to you. You don't want to keep hammering at the same thing over and over because they'll get pissed off and not open up. So you have to have ways that you can figure it out, right?
Yeah, you kind of, although as a journalist, this was one of the hardest things for me to learn when I started out. I did not go to journalism school. I did an internship in the nature offices, which was very helpful, and then figured most everything else out myself. But early on, I discovered, I've got to make a real pest of myself. And it's just part of the job. You talk it through with people. And I've actually found once you do get a hold of a researcher, particularly if you're talking to them about their own work, they're usually pretty willing to take things down to the necessary level and simplify it and talk you through, because they really do want people to understand. And I think particularly in recent years, there's been an even greater appreciation of the importance of that. So, if I do cover a story, I have done a couple of things on nanotechnology and those sorts of physics and engineering fields that I'm just really not well versed in, and I just pick people's brains.
And they don't mind if you go back a few days later and say, I wrote this up, but I don't get this part. Can we go over this again? Usually they're okay with that.
That I've never had a problem with a source recontacting them if it's about their own work.
Yeah, sure.
People are willing to put in the time for that.
Yeah, when it's someone else's, it's less likely.
When it's someone else's, they're not even going to call you back, unless they're a Vincent Racaniello.
Yeah, it's unfortunate. Well, as I wrote online, Rebecca Skloot emailed me a couple of years ago.
Yeah,
and why not? You want to send me your book about helicells? Sure.
Yeah.
Now, if someone did that with string physics, I would say, no, I don't know the field. I can't do it.
Right.
But I figure in your field, virology is a pretty circumscribed field. I can do it. And I'm supposedly knowledgeable enough that it should be easy for me to do. Right. So it's just time in the end.
Exactly. I think really, I wish more researchers would take that approach, and the standard by which they should judge whether they're experts is if they got a paper on this subject from a peer-reviewed journal, would they be really willing to review it?
Yeah, exactly. That's a good point, yeah.
If you would be, if you got a virology paper, you'd be willing to review it.
Sure.
So if you get a reporter calling you about a story on virology, and it's not your work, but it's a virology story, yeah.
Yeah. I find this very interesting, this science journalism interface. As you know, we've had a couple of people on TWiV, Dave Tuller, Trina.
Right.
I think we could do an episode or more on it, getting people to talk about this and how it works, because I'm not sure scientists really understand it.
Right.
I think most scientists feel that they just are rehashing press releases and
sadly, there's a lot of that goes on.
Yeah.
And particularly at some of the print newspapers that are in the process of going out of business, I'm sorry to report, they keep cutting costs, and eventually you've got some sports writer who's now on the science desk, if they even have such a thing.
Yeah.
And they do just rehash press releases. I get all the press releases, and then I see the same wording and the same quotes just smattered all over the place in all sorts of news outlets.
Yeah.
So there is a lot of that goes on, and it would be nice if we could get past that.
Then, of course, there's the headline writers who are totally different, right?
Frequently, yes. For most of the stuff that I do, I'm working for magazines, and in that environment, they'll often take the headlines that I suggest.
Right, but in a newspaper,
the headline writer's somebody else.
Yeah, and
headless body found in topless bar.
Yeah, and they totally change the story sometime, or the conclusions of the story.
Right.
I was reading a story this week. The panda gut microbiome sequence came out, and looking for genes involved in cellulose breakdown, right?
Right.
So my son I was explaining it to him, and one headline we found was, this explains why, pandas can eat bamboo. And if you read the article, in fact, credit to the writer, they said it doesn't prove anything because it just shows that the genes are there.
Right.
Now you actually have to show that the proteins are expressed and that they degrade cellulose. But the headline writer didn't want to do that, right? So you have to be careful. Okay, we have a pick of the week by a listener, Tony from Australia. Here is my pick of the week. I know Vince is interested in online education. This course from Stanford has over 130,000 students enrolled this semester. The two professors are both well known in their field. The online students will be able to take quizzes, exams, and ask questions. The lecturers answer the week's most popular questions. I have enrolled partly because I'm interested in the topic and partly because I'd like to be part of this experiment. So this is a class in Introduction to Artificial Intelligence.
Cool.
This looks cool. You might want to take this, Alan.
Yeah.
I like this. I'm going to check it out because I would love to do a virology course the same way, make it really public. I mean, I put my lectures up, but it's not the same as this where he's actually interacting with the public. So thanks for that, Tony. That looks pretty cool.
Neat.
Okay, TWiV, of course, is found at twiv.tv, where we keep all our show notes and the letters that we read. You can send us email to twiv@twiv.tv. We love to get it. We're also at microbeworld.org, and there you can find an app to stream TWiV to your iPhone or Android device, microbeworld.org/app. Well, Rich Condit is gone. He's still in his meeting, but he's, of course, at the University of Florida, Gainesville. Thank you, Rich, for joining us. You're welcome, Vincent. Oh, that was more like Chewbacca or something.
Yeah.
Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello at virology.ws. Next week, we will do a TWiV in Brazil. I'm heading down there for the National Virology meeting, and I have a couple of Brazilian virologists lined up to talk. So the next time you hear TWiV after this, it will be from near Buenos Aires. No, not Buenos Aires, São Paulo. So that should be a lot of fun. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.